Hello everyone. So we are continuing our series of lectures on the molecular biology, and in this short lecture we are going to talk about two very important topics. One is the Chargaff rule, and the second is the genetic code. Now again, very simple topics, but very frequently asked in exam. A lot of questions from Chargaff rule. Chargaff rule can be somewhat confusing, and lot of slightly correct sounding options in the genetic code. So the genetic code questions tend to be tricky, and the Chargaff rule can be somewhat confusing. Although if you have done them properly, they are very simple topics, and you will score very well. We'll take a look at the questions coming from Chargaff rule and the genetic code. First, let us quickly go through these two topics. The Chargaff rule, applicable for double-stranded DNA. Tells us about the pairing between the nitrogenous bases. I had mentioned in one of the previous lectures that adenine will pair with thymine and guanine will pair with cytosine. Adenine will pair with thymine, guanine will pair with cytosine. That's why the amount of adenine will be equal to amount of thymine. Every time you have an adenine molecule, you have a thymine molecule, so they are going to be equal. Similarly, the amount of guanine is equal to amount of cytosine. Every time you have a guanine molecule, you are going to have a cytosine molecule, so the amount of the two will be equal. If you add both the sides, adenine plus guanine is equal to thymine plus cytosine. In other words, the amount of purines equals the amount of pyrimidines. Now, the Chargaff rule specifies the amount of adenine is equal to amount of thymine, the amount of guanine equal to amount of cytosine, the amount of purine equal to pyrimidine in double-stranded DNA. Right? Now, these information can be extrapolated. Meaning, what do I mean by extrapolation? The adenine is equal to thymine. This means the ratio of adenine and thymine is equal to 1, or the ratio of thymine and adenine is equal to 1. Similarly, ratio of guanine and cytosine is equal to 1, and the ratio of cytosine and guanine is equal to 1. The amount of purine and pyrimidine, because they are equal, the ratio is 1, and the ratio of pyrimidine to purine is equal to 1. We can also say that the ratio of A plus G upon T plus C, which is the purine to pyrimidine, is equal to T plus C upon A plus G. Now, in the question, a lot of permutation combination come. Typically in the PGI exam, they will give you 5 options. Which of the following is correct as per the Chargaff rule, and it becomes very difficult to work out each of the given options because you have lots of permutation combination which are possible. For example, in the last PGI exam, we had one of the options like this: A plus C is equal to T plus G. Is this information correct or not? Now, at the face of it, it doesn't look to be correct. This is not the purines total, is not the pyrimidine total. But if you go into the details, take a look at our rules. Amount of adenine is equal to amount of thymine. The amount of guanine is equal to amount of cytosine. Nowhere it is mentioned that guanine should always be on the right side or should be on the left side. You can write it any way. Both of them are equal. And now when you join the two sides, we find that A plus C is equal to T plus G. Now it becomes very tricky. What to do? There are so many permutation combinations. How many can you work out and how many can you remember? It is not possible. It is not possible to remember all of them. So I'll give you a cheat code. What we do? We label this as rule number 1, and we label this as rule number 2. And now, wherever in the equation these parameters come, we replace it with the corresponding number. For example, if it is part of rule number 1, we write 1. If it is part of rule number 2, then we write 2. For example, A1, T1. Ratio of 1:1 is 1. Okay, this is correct. This pairing is correct. Look at this. A is rule number 1. C is rule number 2. T is rule number 1. G is rule number 2. Meaning 3 should be equal to 2. This is correct. Therefore, this combination is also correct. So rather than trying to memorize and focus on such huge permutations and combinations, just use the number for the reaction. You can make any equation as the reaction 1 or the other one as reaction 2. It is not hard and fast that this will always be 1, this will always be 2. No. You can use any of them as 1 and 2. Replace the values. Compare the two sides. If the two sides are equal, then the given equation is correct. If the two sides are not equal, obviously the given equation will not be correct. The Chargaff rule is very frequently tested in your exam. With the help of different permutations and combinations. A very important additional point which you have to know is semi-conservative replication of DNA is due to the Chargaff rule. Due to the Chargaff rule, we get to see the semi-conservative replication of DNA. I'm sure all of you know that if you have the double-stranded DNA and if you allow it to replicate, only one of the strands is brand new. The second strand will come from the parent DNA. You can see only one strand is new. The second strand is coming from the parent DNA. This is known as the semi-conservative replication. Half of the previous structure is conserved. This is required because we have to follow the Chargaff rule to synthesize the new DNA. We have to do the complementary base pairing to synthesize the new DNA. Therefore, half of the DNA has to be conserved. Otherwise, how will you do the pairing? So, semi-conservative replication of DNA is due to its following the Chargaff rule. Nowhere else will you find this property of semi-conservative replication. Nowhere will you find this property of semi-conservative replication. So, that was about the Chargaff rule. You get to know, you get to identify whether a given combination or a given equation is correct as per Chargaff rule or not. The second part that I mentioned we are going to talk about is the genetic code. We will talk about the genetic code. Now again, this is a very simple topic, but a lot of scope for confusion. First, I'll list the properties of the genetic code and then we'll talk about what we mean by genetic code. We are talking about the properties of the genetic code. The genetic code is a triplet code. The genetic code is a triplet code and it is degenerate. It is degenerate. At the same time, it is unambiguous. The genetic code is non-overlapping. And it is commaless or non-punctuated. And finally, the genetic code is universal. What do you mean by triplet code? 3 nucleotide arranged one after the other will represent one amino acid. 3 nucleotide will represent one amino acid, meaning together the 3 nucleotides will make up one codon. 3 nucleotides will make up one codon. Now we know that each position can have 4 different possibilities. So there are 64 combinations. There are 64 combinations, out of which 3 are stop codons. So you're left with 61 codons. These 61 codons code for only 20 amino acids. They code for only 20 amino acids. This is degeneracy. Meaning codon 1, codon 2, codon 3, all of them code for the same amino acid. Multiple codons coding for the same amino acid is known as degeneracy. But every time the translation occurs, the translation will be the same. It is not possible that at one instance you translate it, you get amino acid 1, and after some time if you translate it, you get amino acid 2. This is not possible. The genetic code is unambiguous. When I say non-overlapping, what do I exactly mean? When I say non-overlapping, I mean if you have the sequence and our genetic code is a triplet code, this means this is codon 1. The same nucleotide cannot be used for another codon. It is not allowed. The genetic code is non-overlapping. One nucleotide cannot be used for multiple codons. Similarly, you cannot skip nucleotides. Meaning, if this is codon 1, then codon 2 cannot start here. You cannot miss the nucleotide. Doesn't matter from where it comes. It comes a mutation, insertion, deletion, whatever the method by which the nucleotide has come. If the nucleotide is there, it must be included in the codon. The genetic code is commaless or non-punctuated. And when I say universal, I mean universal on whole earth. All living form will translate the genetic code with the same results. All living forms will translate the genetic code with the same results. This is very, very important. Now, how do the questions come in your exam? They'll ask you which of the following is not a property of genetic code, and then remove a small part. So your options will become triplet, degenerate, ambiguous, non-overlapping. And if you're not revised, you look at ambiguous and you say, oh, there's something I've heard. This is probably correct. Let's look at the other options. So the options are simple, but they can make it slightly complicated, or you can say they can make it slightly tricky. So please be very careful with the options when you're talking about the genetic code. Please be very careful about the options. Now, sometimes there can be change in sequence of nucleotides. There can be change in sequence of nucleotides due to multiple reasons. There are a lot of ways in which this change can occur. When such a change is seen in the sequence of nucleotides, we call it mutation. Now, there are various different types of mutation, but we have to focus on only two of them. The first is base substitution. Also known as point mutation. And the second is insertion deletion. The base substitution or point mutation is by far the most common type of mutation, and therefore the most commonly asked in your exam. The base substitution is by far the most common type of mutation and most frequently asked in your exam. Let's take a look at the base substitution. If you look at the RNA, the different purines and pyrimidines would be adenine, guanine, cytosine and uracil. We are talking about RNA. Now, if I replace adenine by guanine or guanine by adenine, meaning purine by another purine, or if I replace cytosine by uracil, uracil by cytosine, a pyrimidine by another pyrimidine, this change will be known as transition. This change will be known as transition. But I do something different. A purine replaced by pyrimidine, a pyrimidine replaced by purine, purine replaced by pyrimidine, or a pyrimidine replaced by purine, purine replaced by pyrimidine, pyrimidine by purine, or a purine replaced by pyrimidine, pyrimidine by purine. So interchange. This is called transversion. So you can have transition or transversion when we are talking about base substitution. When we are talking about the base substitution, any of the two can occur. Now, what are the effects of the transition and transversion? What are the effects of transition and transversion? We'll take an example. We'll take an example and see what are the effects of transition and transversion. For our discussion, let's take the example of AGA. You have come across this codon previously when you were talking about mitochondria, where I had said the AGA normally codes for arginine. It normally codes for arginine. So what I'm going to do? I'm going to do a base substitution, replace adenine by guanine. So now our sequence becomes AGG. Now surprisingly, the new codon also codes for the same amino acid. So what has happened? Although there has been a change in the nucleotide sequence, you can see the adenine has been replaced by guanine, but there is no change in the amino acid. Because there is no change in the amino acid, the protein does not get to know. When the protein does not get to know, it does not make any noise. So there is mutation. But the protein does not get to know about it, and therefore it is silent, known as the silent mutation. Let's carry out the same change but in a different place so that our sequence now is GGA. Now GGA codes for glycine. Now this is interesting. The meaning, the translation, the sense was supposed to be arginine, but because of the base substitution, the meaning has become incorrect. It is wrong. There is a mistake here. Because the sense is incorrect, showing a mistake, we call it the missense mutation. Now, sometimes there can be catastrophic results due to a small change. So again, we have replaced one nucleotide, resulting in formation of a stop codon. When you get a stop codon, it does not make any sense, meaning it does not get translated. The stop codon does not get translated, resulting in the nonsense mutation. So the different types of mutation as a result of is in transversion or transition are the silent mutation, the missense mutation, and the nonsense mutation. In case of silent mutation, you get an unchanged protein. In case of nonsense mutation, you get a truncated protein. Truncated protein means the protein has become short. Wherever the stop sequence is synthesized, at that point the translation will stop. So you get a small protein which is called the truncated protein. What happens in the missense mutation? You get a protein with one amino acid changed. We get a protein with one changed amino acid. We're talking about the missense mutation. We're talking about the missense mutation. We get a protein with one amino acid which has been changed. Now, what is the acceptability of this change of a single amino acid? Acceptability of the change of a single amino acid. Various options that we have. It may be completely acceptable. On the other hand, it may be completely unacceptable. We also have a middle ground where we say the change is partially acceptable. The change is partially acceptable. Now, here we should make note. In case of acceptable change, there is no change in physical, chemical, or biological properties. In partially accepted, there is change in physical and chemical properties, but the biological function is intact. The biological function is intact. In case of unacceptable missense mutation, there is loss of biological activity. There is loss of biological activity. We'll take some examples. Example of unacceptable missense mutation is HBM, also known as hemoglobin Boston. This is the inherited methemoglobinemia. Inherited methemoglobinemia, histidine is typically replaced by tryptophan, where the amino acid is unable to maintain iron in the reduced state. It gets oxidized to the Fe3 plus condition, whereby it cannot transport the oxygen. Examples of partially acceptable missense mutation are HBS, C, D, E, O. All of these are examples of partially acceptable missense mutation. About the acceptable form, we have the hemoglobin Hikari, hemoglobin Adelaide, even the Milwaukee form, hemoglobin Milwaukee. I'll write it here. So these are the various different types of the hemoglobin based on their functioning. Please be advised that HBS, most common abnormal hemoglobin variant. So they'll ask a lot of questions about the HBS. They'll ask a lot of questions about HBS. You should know everything about HBS. In HBS, there's involvement of beta chain in the A helix at position 6. Glutamic acid is replaced by valine. Very good. So when you go to exam, you remember beta chain A helix position 6, but the glutamic acid and valine gets mixed. You remember that there was glutamic acid and there was valine, there was some change, but what is there and what has gone, that part has got totally mixed up. So remember, the G is gone. Glutamic acid is gone, replaced by valine. The G is gone. The glutamic acid is replaced by valine. The HBS is very frequently tested upon. The change occurs in the beta chain in the A helix at position 6, where glutamic acid is replaced by valine. This is an example of partially acceptable missense mutation. Partially acceptable missense mutation. To summarize, please note that in case of base substitution, or also known as the point mutation, there can be transition or there can be transversion. Transition means purine replaced by purine, pyrimidine replaced by pyrimidine within the same group. Transversion is purine replaced by pyrimidine or vice versa, means across the group. The change is occurring across the group. As a result of this change, we get to see the silent mutation, the missense mutation, and the nonsense mutation. In silent mutation, we get an unchanged protein. In case of missense mutation, we get a protein with one amino acid changed, and in case of nonsense mutation, we get a truncated protein. Protein is there, however, its size would now be diminished. Take a look at the protein with one amino acid changed, which was the missense mutation. Missense mutation may be acceptable, it may be partially acceptable, or it may be unacceptable. The examples being hemoglobin Hikari, Adelaide, and Milwaukee for the acceptable, the HBS CDENO for the partially acceptable, and the HBM, also known as hemoglobin Boston, for the unacceptable category. Please note, HBS is the most common abnormal hemoglobin variant. It is the most common abnormal hemoglobin variant, whereby in the beta chain in the A helix at position 6, glutamic acid is replaced by valine. The glutamic acid is replaced by valine. So that was the first most common type of mutation. The second, like I told you, is the insertion and deletion. Insertion and deletion. Now here you have to remember a few things. Normally, insertion deletion causes frame shift mutation. Means the reading frame is shifted forwards. The reading frame is shifted forwards. This is called frame shift mutation. When you have a pair of insertion and deletion, meaning one insertion followed by deletion, or one deletion followed by an insertion, then you get temporary frame shift mutation. You get a temporary frame shift mutation. You get a temporary frame shift mutation, meaning reading frame is restored at second change. When the second change occurs, the reading frame will be restored. So if you have an insertion, that will cause the frame shift mutation. As soon as the deletion occurs, the reading frame will be restored. The reading frame will be restored. And third, no frame shift mutation if insertion deletion occur in multiples of 3. Remember, our genetic code is a triplet code. So if you add 3 nucleotides, you are effectively adding one codon. In other words, you are adding one amino acid. Similarly, if you remove 3 nucleotides, you are removing one codon, and there will be again the loss of one amino acid. So insertion deletion normally cause frame shift mutation. If you have a pair of insertion deletion, then you have temporary frame shift mutation. But if insertion or deletion occur in multiples of 3, then there is no frame shift mutation. This is an exception to insertion deletion. If the insertion or deletion occurs in multiples of 3, then the frame shift mutation will not occur. This question has been asked in the PGI exam. Which of the following insertion deletion will not result in the shift in the reading frame? So the answer will be multiple of 3. In the given question, the answer were 3 and 6. Both the options were there. So the answer would be 3 and 6. So this is about the genetic code, and the different common types of mutation. Although there are more other types, there can be translocation, there can be inversion, so like that we have more types of mutation. But these two, first was the base substitution, and the second is the insertion deletion. You must know about this. You must know about. Thank you very much.